بسم الله الرحمن الرحيم وبه نستعين. عنوان المحاضرة Enzymology, General Concept and Enzyme Kinetics. طلبة الفيرست year of medical student كلية الطب جامعة كربلاء، العام الدراسي 2020-2021. تتضمن المحاضرة بعض الأهداف المركزية الرئيسية. First of them, describe the principle of enzyme action, classification, factors affecting their activities, علاقة الانزيمز مع الكوانزيمز or specific vitamin B complexes. أيضا mechanism of enzymatic action, induced fit theory and the lock and key theory, وعلاقة الانزيم inhibitors والtypes of enzyme inhibition. Therefore, role of enzymology or enzyme activity measurement in clinical diagnosis, and then role of enzymology in genetic diseases. Enzymes are proteinic compound or conjugate proteins or nucleoproteins complexes with protein. They are catalysts utilized by essentially all mammalian cells in specific biochemical reactions. Enzymes, the exact role of enzymes is to accelerate or speed up or increase the rate of the biochemical reaction. The exact rule is to increase the rate of the biochemical reaction. The enzyme catalysis affected by different reaction conditions, including pH, temperature, protein and salt concentrations, and presence or absence of organic molecules or inorganic metal ions. All of these are optimal for enzymatic action. The enzyme kinetics allows for laboratory measurement of plasma enzyme levels as well as estimation or calculation of their activity or enzyme inhibition. Damaged cells within organ can release of enzyme into circulation. This enzyme found in plasma may be elevated and they are used as indices of diseases of specific or to specific organ disease and dysfunction. Many enzymes have isoenzymes. Isoenzymes, which are different forms of enzyme, different forms of an enzyme that catalyzes the same biochemical reaction, but they are differ in their physical, chemical, kinetic, and various other properties, and they are either distributed within different in different organs, or may be present within the same organ, but they are catalyzes the same biochemical reaction. As indicated previously, the enzyme acts as a catalyst, that means accelerate or increase the rate of specific biochemical reaction, such as reaction E1, which react with S1, forming the intermediate ES complex, which dissociate, forming the product and the enzyme E1 liberated. The enzyme E1 cannot react with another type of substrate. That means the enzyme is highly specific for the S compound. Example, urease enzyme is specific only for urea hydrolysis to produce ammonia and carbon dioxide and cannot act on another amide compound. Therefore, the enzymes are very highly specific compounds and are very specific for the substrate. Enzymes catalysis is very rapidly. That means only one molecule of an enzyme can convert or act upon about 1,000 of substrate molecule. The substrate is a reactant compound. In enzymology, the reactant is called substrate. Each enzyme can convert 1,000 substrate into product per 1 minute. Therefore, the enzyme catalysis is very rapid. Lack of enzyme will lead to block in metabolic pathways, causing inborn error of metabolism. As indicated, the substance upon which an enzyme acts is called the substrate. The enzyme will convert the substrate into the product or various products. Enzymes are a proteinic compound. Therefore, enzymes follow the physical and chemical properties of protein, such as heat labile and water soluble. Enzymes can be precipitated by various precipitating agents used in precipitation of protein, such as ammonium sulfate or trichloroacetic acid. The enzyme contains about 16-point-something% nitrogen as a constituent atom. The enzyme is highly specific. That means either catalyzed a unibiochemical reaction, one enzyme catalyzed only a unibiochemical reaction, or they are catalyzed various types of substrate conversion into product, such as group specificity. When the enzyme catalyzed a unibiochemical reaction is called absolute. In the other case, called a group specificity. The enzyme also, which are highly specific compound, are stereoisomeric enzyme specificity. That means when we have trans or cis double bond compound, only trans or cis enzyme are required and can convert trans or cis compound into the product. L-amino acid, levo-amino acid, required L-amino acid oxidase in the presence of oxygen to form keto acid and hydrogen peroxide. D-amino acid, dextroamino acid, in the presence of oxygen, catalyzed by D-amino acid oxidase, forming hydrogen peroxide and keto acid. Therefore, enzymes exhibit geometric specificity, وهذه تسمى stereoisomeric or stereochemical specificity as indicated in figure number 1. The orientation around double bond either trans as shown above or below, which is cis. We have trans and cis. Fumarase convert trans double bond, fumaric acid, catalyzed by fumarase, forming malic acid, and cannot convert malic acid, which is a cis double bond compound. The group specificity example, we have phosphatases, which catalyzes the cleavage of phosphate ester bond, esterases, ester linkage, and we have other type of enzyme specificity, which is called reaction specificity, such as conversion of pyruvate. Pyruvate is the end product of glucose catabolism or oxidation. The end product is pyruvic acid. Pyruvic acid may be converted into lactate by lactate dehydrogenase, or pyruvic acid converted into acetyl coenzyme A by pyruvate dehydrogenase complex, or pyruvic acid converted into ethanol by another pathway or biochemical reaction called alcoholic fermentation. Therefore, we have a unique compound converted into various products by various enzyme catalyzed reaction. They are called also absolute enzyme specificity. Therefore, all the enzymes are a proteinic compound. All the enzymes are biologic proteinic compound. Accelerate the biochemical reaction, convert substrate into product. Okay? The chemical nature of enzymes. The enzymes, when they are active, that means act as a catalyst, must be in activated form. The activated form of enzyme is called holoenzyme. The holoenzyme, which is the active enzyme molecule, is equal to the apoenzyme portion. The apoenzyme portion is the proteinic portion of the enzyme molecule. The polypeptide chain of holoenzyme molecule. And another group may be present, which are called cofactor. The cofactor are divided into either coenzyme. The coenzymes are activated form of water-soluble B-complex vitamins. And the other type are metal ions. The metal ions are either negatively charged or positive charged metal ions. Some enzymes are active in the presence of only apoenzyme or protein portion or apoenzyme, such as pancreatic ribonuclease. Other enzymes are composed of metal ions beside the apoenzyme. Other enzymes composed of coenzyme beside the apoenzyme. The large compound of enzyme molecule is due to the presence of apoenzyme, which is large in their molecular weight. The coenzyme is a small molecular weight compound, or they are derived from or activated forms of water-soluble vitamins. Therefore, the haloenzyme may be composed also apoenzyme and both cofactor and coenzymes. When the metal ions or coenzymes are tightly bound with the proteinic portion of the enzyme molecule, they are called prosthetic groups. That means they are tightly bound with the protein portion of the enzyme molecule. The following table indicates the various types of water-soluble B-complex compound with their coenzyme form and their association with a typical reaction type with various enzymatic molecule. For example, the first one, thiamine coenzyme or vitamin B1, which is present in dietary materials, various dietary materials composed of thiamine. The thiamine, after ingestion, digestion, and then absorption, is converted into the activated form, which is called thiamine pyrophosphate. The thiamine pyrophosphate is the coenzyme derived from water-soluble vitamin thiamine. The coenzyme form of thiamine اللي هو thiamine pyrophosphate, this coenzyme associated with various enzymes that catalyzes the decarboxylation reactions. That means decarboxylases. The other type is riboflavin or vitamin B2. Two coenzymes are obtained. The first one, flavin adenine dinucleotide or FAD, and the second is flavin mononucleotide or FMN. Both of them are present as either oxidized form or reduced form with two hydrogens or hydride and hydrogen ions. Both of them are associated with various enzymes catalyzes the oxidation reduction reactions. Also, vitamin B3 or nicotinamide. Two coenzymes are obtained. The first one, nicotinamide adenine dinucleotide, and the second one is nicotinamide adenine dinucleotide phosphate, positively charged. Both of them are oxidized. Also associated with various enzymes catalyzes the oxidation reduction reaction. They accept hydride ion forming NADH and NADPH, and the hydrogen ion is associated with each one. Both of them are present as oxidized or as a reduced form. The other one is pantothenic acid. Pantothenic acid or vitamin B5, the coenzyme derived from pantothenic acid is coenzyme A. Coenzyme A acts as a carrier in various metabolic pathways, carry a metabolite through a metabolic pathways. Pyridoxal vitamin B6, the coenzyme obtained is called pyridoxal 5-phosphate. The pyridoxal 5-phosphate associated with various enzymes catalyzes the transamination or deamination reactions of amino acid. The biotin, the coenzyme obtained from biotin, when biotin is linked covalently with lysine amino acid, the lysine is an amino acid residue present within a polypeptide chain of enzymes, various enzymes called carboxylases. When biotin linked with lysine of carboxylases, the obtained compound is called biocytin. Biocytin is the coenzyme form of biotin associated with various carboxylases. That's mean addition of carbon dioxide. The other one is folic acid. Tetrahydrofolate is the active form. Tetrahydrofolate act as a carrier of one carbon unit, such as methyl, methylene, methinyl, or formamino or formyl. All of these are one carbon group transferred by tetrahydrofolate through the metabolic pathways, such as in purine-based biosynthesis, in DNA biosynthesis, and so on. Cyanocobalamin, the coenzyme form obtained from cyanocobalamin, is 5-deoxyadenosyl 5-deoxyadenosyl cobalamin associated with 1,2 shift or transfer of methyl groups and rearrangement within the same molecule called intramolecular rearrangement. The ascorbic acid, the last one, ascorbic acid, is water soluble, non-B complex, act as antioxidant. The coenzyme may be simple protein or simple. The enzyme may be simple protein or complex enzyme containing a non-protein part called the prosthetic group, which are called coenzymes. The protein part of enzyme is named apoenzyme as mentioned, and the active form is called haloenzyme. Coenzymes may be of two different groups. One of them catalyzes the oxidoreductases by donating or accepting hydrogen atoms or electrons. The other coenzymes taking part in reaction transfer ring group other than the hydrogen. The metal ions and coenzymes, the presence of metal ions increase the activity of the enzyme. If the metal ions or activators are absent, no enzyme activity takes place, such as zinc in alcohol dehydrogenase, iron in cytochrome oxidase, molybdenum in xanthine oxidase. When they are absent, no enzyme activity is obtained. Metalloenzymes contain highly tightly bound metal ions. The metalloenzymes mean the metal ion is tightly bound with the protein portion of the enzyme molecule. That means the metal ion acts as a part of the enzyme, total enzyme molecule. Therefore, deficiency of the metal ion in metalloenzymes indicate the enzyme is inactive. Okay? The metal ions in metalloenzymes are tightly bound within the protein portion of the enzyme molecule. The other type of enzyme and metal ions is the metal activated enzymes, which contain weakly or loosely bound metal ions to the enzyme, such as sodium, potassium, calcium, and can remove by dialysis. Even without the metal ion, the enzyme may be active. But when the metal ion is added, the activity is enhanced. They are called ion-activated enzymes, such as calcium ion will activate pancreatic lipase. Some prosthetic group are metallo-organic compound, such as heme, magnesium Mg plus 2 for phosphortransferase with inactive without magnesium ion, the enzyme is inactive. Manganese is the metal ions for arginase. Without the manganese, the arginase is inactive enzyme. Peroxidase, catalase required ferric ion, ferrous ion, and so on. Potassium for pyruvate kinase. Okay. The coenzyme or metal ions may act as a co-substrate, as an organic factor that behaves as a second substrate in an enzyme catalyzed reaction. The co-substrate may be a coenzyme such as NAD plus can act as co-substrate for many oxidoreductases. Enzyme active site. What are the active site? The active site of the enzyme molecule is a region that bind substrate and convert it into product. A region. Small part of whole enzyme molecule. Substrate binds in active site by multiple weak forces or chemical forces such as electrostatic interactions, hydrogen bonding, van der Waals interactions, hydrophobic interactions, reversible covalent bonding. All of these chemical forces bind the substrate with the active site. Therefore, binding of the substrate to the active site give the enzyme substrate complex. When the substrate bind with the active site of the enzyme substrate complex is produced as indicated below. This figure number 2 indicate the enzyme molecule, where the enzyme molecule as indicated below in black composed of the active site. The substrate is bind with the active site, which is complementary for binding with the active site, forming the enzyme substrate complex as shown in the second or first intermediate. The enzyme substrate complex, in this complex we have chemical forming, and then the enzyme product complex is produced, and the product leave, or this complex enzyme product complex dissociate, forming the product of the biochemical reaction, and the enzyme is liberated as unchanged molecule. Therefore, the enzymatic reaction is a biologic catalyst, accelerate the biochemical reaction, forming a product, and the enzyme leave unchanged. The enzyme catalyzed reaction as are indicated in this box or figure. Substrate react with the enzyme molecule forming ES complex. Within the ES complex, the reaction occurs to convert substrate to product, forming enzyme product complex, and then this enzyme product complex dissociate, forming the free enzyme and the product. The product which are no longer attracted to the active site are released. The overall is conversion of the substrate into product by the presence of enzyme. How the enzyme catalyzed reaction? Two models, we have two theories. The first theory is lock and key theory. Lock and key theory or موديل نظرية المفتاح والقفل. The active site is complementary to the substrate binding. This complementary arrangement is referred to as lock and key fit of the substrate in the active site, as shown in a previous figure and in the next figure indicated below. This is a lock and key theory. Lock and key theory or model the active site of the enzyme as shown. B is complementary for substrate binding, forming the D complex. The D complex هو الانزيم substrate complex which dissociate, forming the product and the enzyme leave. This is the lock and key model. The other model is induced fit model, as indicated, is not complementary substrate binding with the active site. Not complementary, لا يكون متمما. Sometimes the shape of the substrate molecule does not exactly match the active site or the shape of the active site of the unbound enzyme is not the exact complement of the shape of the substrate. The enzyme does not bind to substrate. After binding of the enzyme to the substrate is initiated, a conformational changes in the shape of the active site which result in a new shape of the active site that is complementary to the shape of the substrate. Active site of induced fit enzyme only become complementary after substrate is bound. اوكي وهذه عندما تكون الاكتف سايت not complementary وتزداد تراكيز المواد الاساس، هذه تضغط على الاكتف سايت ليكون متمما للسبستريت، وبالتالي التفاعل يحدث ويحول السبستريت الى برودكت في ظروف وقتيه محدده. The exact role of the enzyme. What is the exact function of the enzyme molecule? Enzyme lower the activation energy required to convert all molecules of reacting substance from the ground state to the transition state. Substrates are remaining in an energy trough and are to be placed in at a higher energy level where upon spontaneous degradation can occur. For example, suppose we want to make a fire. يعني واحد ممكن أن يعجبه يشعل فرد نار fire. Even if we keep a flame, the wood will not burn initially. We have to add kerosene حتى نشعل. انت عندك هاي الجداحة وعندك حطب for example، راح نضيف الكيروسين or paper to initiate burning، نبدي الحريق الهائل وتوليد الطاقة الكبيرة. بنفس المعنى the activation of energy is to be initially supplied. During enzyme substrate binding, weak interactions between enzyme and substrate are optimized. This weak binding of interactions between enzyme active site and substrate provide the major driving force for the enzyme or for the enzymatic catalysis. The enzyme in general reduce the magnitude of this activation energy. For example, activation energy of acid hydrolysis of sucrose is about 26,000 calories per mole, while activation energy is only 9,000 kilocalorie or calorie per mole when hydrolyzed by sucrase, as shown in figure number 2. Figure number 2 indicates how the enzyme decreases the activation energy. As shown, enzyme convert substrate into transition state, which carry a higher energy content. In this transition state, we have chemical transformation that occur, forming a product and the enzyme leave unchanged. The change in Gibbs-free energy between substrate and the product, this change in Gibbs-free energy, when this change is in negative value, indicate that the reaction proceeds spontaneously. But when the change in Gibbs-free energy is equal to 0, this means that the reaction is at an equilibrium state, and when the Gibbs-free energy change of Gibbs-free energy is in a positive sign, this indicates no reaction is produced, no reaction product is produced. Okay? The enzyme, the exact role of the enzyme is to decrease the activation energy حتى يوصل طاقة التنشيط بأسرع وقت بأقل طاقة نحتاج إليها. أوكي؟ وبعد طاقة التنشيط راح التفاعل يفقد هذه الطاقة ويكون الناتج، والإنزيم مقدار الطاقة اللي تبقى بالإنزيم تبقى من دون أن تتغير. أعيد الكلام. Substrate, as indicated, convert into product. First, we initiate this conversion. That means the substrate react with the active site, forming enzyme substrate complex, which is in transition state condition. Okay? In this state, transformation reactions occur, forming the product that produced, and the enzyme leave unchanged. Okay? The exact role of the enzyme is to decrease the amount of activation energy to produce ES complex. Without the presence of the enzyme, as shown, delta G is higher than that required by enzymatic reaction. The activation energy is the energy required to convert the substrate into an activated state that is called transition state. يحمل طاقة عالية. هذه الطاقة راح تشجع أو تساعد عمليات كسر أواصر، عمليات بناء أواصر وتكوين نواتج، والناتج يخرج. ومقدار change in Gibbs free energy when in a negative value indicate that the reaction will proceed spontaneously تلقائيا. وعندما يكون مساويا إلى الزيرو، الخط الأزرق هذا البلو، عندما يكون الناتج الخط الأزرق، that's mean delta G is equal to zero. That's mean the reaction is at equilibrium. عندما تكون delta G أعلى من الخط الأزرق indicate no reaction, or the change in Gibbs free energy is in a positive sign. That's mean no reaction occur. في كل التفاعلات الكيموحيوية the reaction must be proceed spontaneously نحو تكوين الهدف اللي هو البرودكت، أي أنه يكون بإشارة سالبة or change in Gibbs free energy is in a negative value. Okay? The enzyme, the exact role of the enzyme is to decrease the activation energy. Now في المثال بسيط أعطيكم وأختم الموضوع أو المحاضرة الأولى. The enzyme catalyzes the biochemical reactions. The enzymes are proteinic compound or biologic catalysis or catalyst increase the rate of the biochemical reaction by decreasing the amount of activation energy. Okay, all of the biochemical reactions must be proceed spontaneously لأغراض تكوين النواتج وإدامة الحياة. لو التفاعلات ما تحدث أو عندما تكون قيمة الدلتا جي in a positive sign تفاعلات لن تتم، therefore الحياة راح تنتهي، وبالتالي change in Gibbs free energy must be in a negative value for all biochemical reactions. هنالك تفاعلات coupling reactions عندما نحتاج to proceed certain type of substrate إلى نواتج، نحتاج إلى أن يساعدنا تفاعل exothermic، وهنالك تفاعل endothermic، وبالنتيجة تكون قيمة change in Gibbs free energy in a negative value to proceed the reaction into the product produced. Okay? هذا ما يتعلق بالمحاضرة الأولى اللي هي بعض المفاهيم المهمة بموضوع الإنزيمولوجي، وبعد ذلك أبدأ بالمحاضرة الثانية اللي هي بعض العوامل المؤثرة على نشاط الإنزيمز والإنزيم كلاسيفيكيشن وإلى آخره. وشكرا جزيلا.